The title of this presentation is Approximating Binding Affinity of Compounds from CellTalkDB Sequences with AutoDock Vina. So first, before we provide a presentation overview, I wanted to provide a few motivation slides describing what would be the interest and what we would help to achieve with this type of computational work. So first of all, we can recognize that molecular dynamics simulations are relying upon predicting the rates at which two compounds interact with one another. And if we're interested in looking at candidates for different compounds that can be used as input from these simulations, we can either look to publicly maintained resources such as the protein data bank or other collection of deposited amino acid sequences that we can use to constitute or to make models that we would like to perform computations on. And with these two considerations in mind, it continues to remain of interest to compare how different molecular docking packages compare to each other. And along these efforts, we're interested in first looking at how AutoDock Vina would perform from some collection of sequences from a publicly maintained database that I'm going to describe and motivate to you. So this is an overview slide, which is going to be describing many parts of the presentation and some of the topics that we'll be discussing. So it's an overview in that we want to discuss results obtained from one open source molecular docking package, which is AutoDock Vina. And this is important for our discussion of molecular dynamic workflows because it draws our attention and it brings several questions to mind relating to what are the types of input files that we would need for running these simulations, what public databases or what other public collection of sequences can we use and leverage for making compounds that we can examine more closely in AutoDock Vina, and how many atoms appear in the PDB-QT files, which is one particular file format that we convert to when we're doing simulations in AutoDock and in AutoDock Vina. And how do we define the region about the active site that the molecular docking or the molecular simulation algorithm will examine to form approximations of binding affinity that we'll mention several times. And finally, how do we interpret the results that we get from the command line and from these algorithms? And with all these considerations in mind, after we're able to implement simulations at AutoDock Vina, we can then ask ourselves, what do the output of the results from the simulations mean, which is along the lines of how do we interpret the results that we can gain from AutoDock Vina, as well as which compounds would be the best or of interest to further examine in the future if we would have unlimited or more extensive computational resources. So we can orient ourselves first with AutoDock Vina by making use of some types of documentation and examples which are provided directly from the developers of AutoDock Vina themselves, in which they provide a video demonstrating how one can make use of either the Python molecule viewer or other types of software packages like PyMol, which will allow us to examine the active site and hence make judgments about the binding activity and the binding affinity of different ligands and receptors together when we define a certain region of space that the molecular docking algorithm will examine. And in this case, this demonstrates a type of method that we use for the compounds I'm going to present results on later in the presentation, and that the way that we set the binding region and the domain region that the algorithm AutoDock Vina examines to form approximations of the binding affinity is that we put restrictions on the total number of points that the algorithm can examine in the X, Y, and Z directions, as well as centering a grid box about however many points we choose in each dimension so that the algorithm can assess and make predictions about different conformations of the ligands and receptors that are going to interact with one another or that are going to be interacting about this active site. And to further orient ourselves with AutoDock Vina, it's also important to think about what is the difference between AutoDock and AutoDock Vina, and how these two different workflows can be married together for actually being able to, which will allow us to actually look at how different compounds can interact with one another. In this plot, it's a plot of the runtime for different compounds that are either compiled in AutoDock or Vina, and the red and the yellow bars in this plot demonstrate how changing the number of CPUs it can result in like a 8-fold reduction in the runtime that AutoDock or that Vina will use for looking at how these compounds would behave and interact with one another. But in the blue bar that's given in the top here, it demonstrates that just running AutoDock without looking at the performance of Vina, it can result in a much longer runtime of the algorithm. So it's just demonstrating how there can be different algorithms which can be used in conjunction together to obtain a reasonable polynomial runtime algorithm that will allow us to characterize the behavior between these ligands and receptors, several of which I'm going to provide specific examples of and continue motivating to you. And besides what we've been talking about AutoDock or Vina and how we're going to make use of CellTalkDB, which is referenced in a publication that we're going to include within the next couple of slides, there can be several motivations for looking at public repositories of sequences. On Twitter, there was a very helpful diagram that demonstrated how from OmniPath, which demonstrated how different types of cells can interact with one another. If we look at the type of graphic that's provided in this visualization that's provided on this slide, we can see that each one of the nodes of the graph is represented by different types of cells, whether it be fibroblast 1 or fibroblast 2, or different macrophages or NK cells and the like. And we can see that we can constitute and draw an edge in the graph between any one of these nodes, which can indicate to us interactions between different cells in this network. So it's really interesting because we can just demonstrate different types of interactions and exhibit the directionality of the edge, in which we can see that for the macrophage node, which has approximately 1,300 cells active, any one of the macrophages which interact with any one of the other neighboring nodes, which can be indicated by any one of different types of cells that are given in this graph, is indicated by a directed black edge that's pointing outwards from the macrophage vertex in this graph, which can help us have a more clear understanding of how different types of receptors or compounds interact with one another. So along these lines, as we've been alluding to, we want to think about the required input files that we would need to have for running AutoDock Vina and this molecular docking software that we've been describing. And we can make use of the CellTalkDB resource, which is available to us for academic and non-commercial use from authors in the Chinese Academy of Sciences, in which in this journal article, they talk about CellTalkDB, which is a manually curated database of ligand receptors interactions in humans and mice. So this is interesting for us because it can give us a broad collection of ligands and receptors that have already been curated and can relate to publicly deposited amino acid sequences that we can use and leverage to test the performance of AutoDock Vina, and to ask, as we had mentioned in the overview slide, how these interactions or how we can predict how these interactions between different ligands and receptors would be under using AutoDock Vina. And the CellTalkDB GitHub provides us with a lot of interesting information that can help us leverage these sequences that are available for non-commercial academic use. For one, in this data collection column that we're indicating on the left-hand side of the slide, we can see that they try to, through their manual curation of different sequences, they try to look at different protein-protein interactions, and then they constitute, or to characterize how these proteins would interact with one another more clearly, we can have some annotation information, whether it be from the UniProt KB repository, which is, as it's described here, it's a protein knowledge base, as well as the NCBI gene or the ensemble. And for the data processing step, once we've collected and aggregated all the data of interest that we want to look at, is that we can remove some duplicate protein interactions in our network, and we can look at how different ligands and receptors could be matched up to each other, which could be good candidates for either human or mouse models of interaction. and looking at these receptors of ligands and or looking at these pairs of ligands and receptors, it can be helpful for us because if we have some knowledge about a mouse model and how disease is propagated and how a certain disease behaves in a mouse model, it can help us leverage this type of knowledge for a human model setting, even though the human model setting and how these ligands and receptors interact with one another can be much more complicated. And for some other columns that are described in the CellTalkDB GitHub, they also provide an overview of the manual verification of these different amino acid sequences and how they are manually checked by 3 reviewers to determine the connections between published texts that have been accepted in journals and how these different ligands and receptors have already been characterized in some properties of the types of receptors in different areas of the genome or different diseases or different contexts in which these compounds could interact with one another. And when it's all said and done, the CellTalkDB database that they provide for us consists of approximately 3,400 ligand receptor pairs for human models, a little more than 2,000 ligand receptor pairs for mouse models, and they also provide a further breakdown of the specific number of ligands and receptors for the human and mouse models. And this is available on their website, which I'll also further describe to you as we motivate and continue discussing the types of compounds from the CellTalkDB database that we can run AutoDock Vina simulations on. So this is an overview of their website, in which it provides, as I had alluded to in the previous slide, an overview of different ligand receptor pairs for mouse and human models. And the whole idea about this is to bridge how the types of input files that they provide from the PDB files can be leveraged to obtain PDB-QT files, which can then be fed into AutoDock Vina for analyzing activity around different binding sites or active sites between these ligand and receptors that they provide in the database. And if we look a little bit more on their website that's given in the CellTalkDB website, we could see that there are different lists that we can browse for ligand receptor interaction pairs. And if you look at any one of these lists that are provided on the CellTalkDB website that they introduced in the CellTalkDB paper that we gave earlier, they give us a lot of interesting detail about ways in which the ligand and receptor pairs can interact and the associated function of each one of these ligand receptor pairs. And this is of interest in at least determining more specific characteristics of how these ligands and receptors interact with one another. And from the CellTalkDB website, as we've been mentioning from the browse section, we can look at and obtain full-length amino acid sequences that are found on UniProt, as well as a corresponding description of the function of these different ligands and receptors. In this particular case, from one compound that's deposited on the CellTalkDB website that was manually curated by the creators and the people who maintain the CellTalkDB website, they looked at this particular type of protein, which is a receptor, is corresponding to some AGTR2 gene, and we can have the total length in amino acids, as well as the some knowledge of the protein existence, which the UniProt database says is evidence at the protein level. And we can also see that the associated function is that it can have a particular role in signaling in this independent pathway that they describe and provide a description of, as well as the organism. So from this UniProt entry and how it's deposited, we can also download the full-length amino acid sequence, which we can use to look at and further examine additional pre-processing steps in our workflow that we can use to run this sequence with another compound of interest in AutoDock Vina. And this is just demonstrating some types of additional characteristics from the TXT files from the CellTalkDB website and the UniProt website, in which from this TXT file we can have a gene ensemble ID, which is given in the leftmost column of this TXT file, as well as the receptor and the gene symbol and the ligand gene ID and the receptor gene ID. And this provides us with a wealth of information because we can have an idea about how many amino acids are contained or how many components are contained also in the sequence that we would like to run AutoDock Vina on, as well as different combinatorial options for different combinations or pairs of ligands and receptors that we can look to examine further in our simulation efforts. And so once we have the PDB files, which are given in AutoDock Vina, or that we want to process further in AutoDock Vina from the UniProt website, which we gain information on from the manually curated selection of sequences or of ligands and receptors that are given in the CellTalkDB database, we can construct a homology model with the Swiss modeler. And this is a very helpful resource because it's able to make the jump or the inference between the sequence to actual 3D structure and how we can be able to feed these PDB Qt files, which are generated from the homology models, into AutoDock Vina for further computational processing. And this is just demonstrating how you can either run this through an account or you can just do it completely open source, and how they also provide some documentation which further describes some of the steps that I'll be outlining when we're running the homology models to obtain a three-dimensional representation of the ligands and receptors that we're interested in looking at with AutoDock Vina. So this is one example of running the output from the Swiss model on one ligand receptor pair from CellTalkDB, in which what we do is that we provide, as we had mentioned, the full-length amino acid sequences, which we can get from the UniProt website that has information that's connected from the CellTalkDB website. And from this, what the Swiss model does to generate different types of homology models is that it generates a total list of templates from which it generates a total number of models. And we can see that from this instance that we ran for generating a homology model in May 26th of earlier this year, we can look at 50 possible templates, as well as 6 potential models that are generated from those 50 templates that the Swiss model believes with high certainty and with high probability would be able to constitute a three-dimensional structure of a ligand or receptor that we can then feed to look at the three-dimensional interactions of to further characterize the binding affinity in AutoDock Vina. And after we run the Swiss model on the input PDB file, which is related to the full-length amino acid sequence that we get, the project compiles the list of the possible models from this fixed number of templates. But to describe further what goes on under the hood, under the Swiss model to obtain this three-dimensional representation that we've been mentioning, is that we can also look at different characteristics of each one of the models that are outputted from the fixed number of templates. Typically, we choose a model which is produced from the list of all possible models, which has the highest sequence and identity and also the highest GMQE score. And this GMQE score is based on training a perceptron, which is a particular type of neural network that appears in many areas of math. So what we look at generally is that we want to look for a high perceptron score as well as a high sequence identity. And this will indicate to us that the Swiss model was able to generate a reliable three-dimensional structure of the compound that we would like to further characterize the binding affinity and the binding affinity or the binding approximation of with AutoDock Vina. So this is just demonstrating that if we have a high GMQE score as well as 100% sequence identity, it's probably best to just go with that model because it will incorporate all sections of the amino acid sequence that we fed into the Swiss model from the UniProt website. And in addition to having chosen a model based on the list of all possible templates and the sequence identity and the perceptron score, the Swiss model also provides a three-dimensional representation of this compound structure that we can rotate and look at to determine different putative or possible sites of interaction that a ligand can have with a receptor. In the case of another compound that I ran on the Swiss model before obtaining the PDB-QT file that we fed to AutoDock Vina, we can look at 16 templates from which 4 models are introduced. And we can look at, as we mentioned, the sequence identity, which again, in this case, for this particular model, 01 was 100%. as well as a GMQE score, which is 0.77, which is pretty good. So what we look at from this three-dimensional representation of the compound is that there can exist different types and areas that we can set the binding docking window of, so that we can look at the type of activity that a certain ligand and receptor would have when we're running our approximations of the binding affinity. So even for another compound that we ran the Swiss homology model on, there can be a substantial difference in the predicted structure of the compound depending on the GMQE perceptron score that we had listed and been describing in the previous slides. For instance, we can see that there can be some substantial difference in either the sequence identity or the GMQE score. And what's interesting and what we have to keep in mind when we're looking at how the GMQE score and the sequence identity work together so that we can obtain a good three-dimensional representation of our compound is that we can look at the GMQE score, which is 0.45, with approximately 81% sequence identity, and how even though this compound from the homology model that we generated with the Swiss model has a higher sequence identity than this other model that's listed below, which is around a little above a 78% sequence identity, the GMQE score, which is a perceptron-based score, is a lot higher in this case. And this is important because if we look directly at a very scaled out, a broad view of Model 1 compared to Model 2, Model 2 has a lot more structure along this part in the tail of the ligand of the receptor that we're looking at in comparison to this compound, which is a lot smaller. So even though the Swiss model, it generates different types of putative or possible homology models that could work for different compounds, it's very important, and as a caveat, also it's important to take both the percentage of the sequence identity as well as the GMQE score into account when choosing which model we would like to use to convert to the PDB-QT file. And this is just demonstrating a three-dimensional view of the model 01, in which one can see that there's a lot more structure, three-dimensional structure, that's predicted for this compound, even though the sequence identity is a little bit lower in comparison to the model 02, which has a GMQE score of 0.45. And we can repeat this procedure to generate homology models for a variety of compounds and ligands and receptors for manually curated items in the CellTalkDB, the CellTalk database that we have been discussing. So for instance, this is just demonstrating many other compounds that I haven't ran AutoDock Vina simulations on, but that I do have the PDB-QT files for, with the information that's given from the open source sequences and the CellTalkDB and the Uniprod databases, by making use of the Swiss model to bridge the gap between the PDB-QT and the PDB files and the three-dimensional structure of each one of the compounds that we're interested in looking at further. So this is just demonstrating that there exists a wide variety of compounds to which these computational methods can be applied to, and how in each one of these compounds, it can be complicated to determine from the morphology and different characteristics of the ligand and receptor which types of areas of interaction would be interesting to examine, and how the algorithm would perform depending upon the region of the binding window that we set upon, or that we set along each one of these possible compounds. And the list of possible homology models goes on. At the end of the presentation, I'll provide a reference to a PDF on my GitHub, which provides more detail as well as several names for the different receptors and ligands that we generated the homology models on that we've been discussing. And now we can actually get to some more detail about the AutoDock Vina implementation and how different steps of the AutoDock Vina algorithm can be executed once we have information through the input and the output PDB-QT files, as well as the size of the binding region that we want to run the algorithm on, which is measured in angstroms cubed. And so this is just demonstrating what appears in the Linux environment when one runs AutoDock Vina, in which it makes use of this 2010 reference in which they first introduced AutoDock Vina by drawing upon elements that we had mentioned earlier in the presentation following the overview slide. And then there can be different ways to set the total volume that AutoDock Vina looks at. But in our case, oftentimes it's very interesting to set the search volume to be a little bit wider so that the algorithm can determine how many combinatorial possibilities there are for looking at how many conformations can be predicted from the structure of the ligands and the receptors. And in particular, in this case, there are certain characteristics of the workstation and the work environment that have to be taken into account when AutoDock Vina is carrying out its computations and its workflow, including the number of CPUs, how long the search volume is in angstrom cubed, the complexity of the PDBQT file input that's read, as well as having to set up the scoring function so that AutoDock Vina can compare systematically different types of conformations and the corresponding binding affinity approximations, as well as the root mean square distance of each one of the binding affinity approximations, which we'll provide some examples of later in some future slides. And then it's going to actually refine the results in which it is able to order and characterize depending upon the number of amino acids that are in contact in the ligand and receptor, so that we can obtain a PDBQT output file that has its own three-dimensional structure that we can look at for further examination. And this is just demonstrating the implementation steps that we have been describing. But briefly to reiterate, because they'll show up and appear in the two output files and the two examples of output that I'll demonstrate to you, is that we define a search volume, which is dependent upon the number of CPUs. And then we load the input, which is relating to a scoring function, as well as looking at the binding site. And after performing a search around the binding site, the results are compiled to 100%, from which the PDBQT output file is produced. And this is demonstrating how different types of conformations can have several types of binding affinity. And besides the previous steps of AutoDock Vina that I highlighted to you and described, after the results are refined, we can look at two different examples, which are not ran on CellTalkDB sequences, but other sequences of interest, which can demonstrate different types of modes that we can get for countably many or however many conformations AutoDock Vina produces, as well as the binding affinity, which is measured in kilocalories per mole, as well as the root mean square distance. So these are just two numbers from which one can form interpretations about the orientation of one ligand and receptor relative to each other when the simulation is being ran. And one can see is that what's important to notice in AutoDock Vina is that actually the negative binding affinities are actually very good to have, and they indicate that it's more likely for this ligand and receptor to interact. And one can see that the binding affinities in the command line of the Linux environment, they're ordered from as you can see, minus 6.6 to minus 3.6, so from least to greatest, as well as even here, a binding affinity range of minus 7.2 to minus 5.5 is listed over 9 possible conformations. And we can further describe what type of output we get from AutoDock Vina by looking at one compound of interest from the CellTalkDB repository, in which this compound of interest is related to the ADA2 receptor. And the question that we would like to look at and further examine is how many conformations would we expect to obtain from this computational chemistry workflow from AutoDock Vina. And this is demonstrating several different types of perspectives of the three-dimensional structure that we can get from the ADA2 receptor, as well as the corresponding ligand, which is listed and provided to us on the CellTalkDB website. And from the information that's provided in the database, on the CellTalkDB database, we can see that there can be different types of interactions between this ligand and receptor, which involves a very substantial interaction along this ternary structure, which is given by the overlapping region between this ligand and receptor that's provided. And from from different types of molecule viewers that we had listed earlier, like the Python molecule viewer or PyMol. We can look at a three-dimensional representation and look at different perspectives to gain further insight about how these different ligands and receptors would interact with one another, and how different types of binding affinity approximations would be produced from this AutoDock Vina workflow that we have been describing. And this is just demonstrating the homology model of the ADA2A receptor, which is provided from the Swiss homology model that we use as a PDB file that we can leverage and make use of to get the PDB-QT input file that's required for AutoDock Vina, so that we can run the simulations for determining an approximation of the binding affinity. And we can also further describe, before we provide the actual numerical approximation of the binding affinity for one of the compounds, another potential compound of interest relating to the ADA2A receptor, or I'm sorry, the TNR6 receptor, which is provided in the CellTalkDB repository, in which we can ask the question about how many confirmations would we expect to obtain from the AutoDock Vina workflow for a different compound of interest. And there can be several different types of characteristics of the three-dimensional structure of these compounds, which can be provided from this receptor in comparison to the other receptor. If we compare these two receptors side by side, the TNR6 receptor in comparison to the ADA2 receptor that we provided and described in the previous two slides, it's given by a compound that's or a ligand and receptor pair that has a much shorter amino acid sequence. And if we set a large enough binding region about the active site, allows us to generate 9 possible conformations, which can be obtained by rotating the ligand given a certain action, or in a very controlled way, given a certain angle, to obtain different conformations about this TNR6, about the ligand, so that we can obtain different types of possible binding affinity interactions about the active site that we have defined. And this is demonstrated very clearly by different types of three-dimensional representations of each one of the possible 9 conformations that AutoDock Vina produces, in which we can look to color, either in yellow, red, blue, or green, different types of conformations, and how they are positioned relative to the central object, which is the ligand, or which is a receptor, in different types of orientations that the ligand can assume when interacting with this receptor to obtain some binding affinity approximation. And this is just, again, providing, as we did in the previous case, an image of the TNR6 receptor in the homology model that we obtained from the Swiss homology model to obtain a PDB file that we used to obtain a PDB-QT file that was fed into AutoDock Vina in the corresponding 9 conformations that we had produced and described in the previous slide. And after setting the parameters appropriately for the active site between the interaction between the ligand and receptors, we can obtain the following results. So now we described how the different conformations can be produced from AutoDock Vina, how we obtain the input files, how we set the binding region so that we can examine the activity about the active site and the approximated binding affinity, as well as the output that's provided from running AutoDock Vina, the AutoDock Vina workflow on the receptor and ligand PDB-QT files, as well as specifying 6 other parameters besides the output PDB-QT directory. And this is just demonstrating that we only were able to produce 1 conformation, and that this was a very computationally intensive conformation for AutoDock Vina to approximate. And it's a positive value, indicating that even though the algorithm can predict that there can exist a very substantial interaction in the ternary structure between the ligand and the receptor and how they interact approximately in this fashion, that actually, probabilistically speaking, even though such an interaction could occur, it could not be as likely as the interaction which is given from the output in AutoDock Vina that has a negative binding affinity, which is measured again in kilocalories per mole for the other compound I'll describe to you in the next slide. And so this is describing the results from AutoDock Vina for the second receptor, and this is demonstrating how the 2 receptors and running AutoDock Vina on them can be juxtaposed and how the output of the algorithm is different from each one of these cases. In this case, as we had alluded to previously, a negative binding affinity that's produced by AutoDock Vina, which is measured in kilocalories per mole, also indicates that this interaction is more likely to occur. And in this case, we can see that there are 9 binding there are 9 modes or 9 conformations which AutoDock Vina produces, which we can look at here, which range from negative 15.7 kilocalories per mole to negative 14.4 kilocalories per mole, as well as different values for the root mean squared distance. If we look at and compare different values, we can see that conformation 2 as well as conformation 6 have very similar values for the two values of the root mean squared difference, but that the corresponding binding affinity, which is measured in kilocalories per mole between each one of these two conformations, differs a little bit. So this can help us establish, if we look at these results in the Python molecule viewer from the output PDB-QT file, how these different types of interactions would look and how they would be represented in three-dimensional space, and which type of interaction we could think is more likely to occur, or is more feasible to occur if these ligand and receptors were to interact with one another. So I wanted to end the presentation with some conclusions, which can describe broadly what we have been speaking about. So what we can look at is that we motivated current interests, which are both in academia and in industry, for simulating behavior between ligands and receptors. And after further discussion beyond this motivation, we introduced CellTalkDB. And as a public resource for academic use, CellTalkDB provides many combinatorial arguments or targets that we can investigate, including human and mouse receptors and corresponding ligands. And we can further build upon the resources that are provided by CellTalkDB, in which we describe results generated by AutoDock Vina for 2 ligand receptor pairs. And as I'm going to include as a link to my GitHub in the description of this video, I also provided several additional compounds of interest whose homology models are provided in this PDF that are of interest for future simulations and computationally related efforts. And yeah, thanks for listening.